Here in part 4, let's explore mitochondria and chloroplasts further. One important thing to recognize is that the size of prokaryotic cells is limited by diffusion. Prokaryotes, being small, benefit from having a very high surface to volume relationships. But eukaryotes do not have this same surface to volume relationship because eukaryotic cells tend to be larger. So prokaryotic cells are about a micrometer in diameter. And if we go from the typical size of a bacteria to that of a eukaryotic cell, we see that the surface to volume relationship changes by a factor of 10, from a surface to volume ratio of 3 down to one of only 0.3. The advantage in a prokaryotic cell is that no internal part of the cell is ever very far from the surface. So when diffusion is needed to bring in materials, to make them available at the surface of the cell, this can quickly happen with a high surface to volume relationship. So if the eukaryotic cell is derived from a series of engulfments, why did it arise only once? Well, one idea is that the invention of the eukaryotic cell, the evolution of the eukaryotic cell, was a rare breakthrough, and that it transcended a significant barrier in terms of energy generation. Recall, as I just said, that as a cell gets larger, its surface to volume ratio decreases. Yet, we need membranes that are within the prokaryotic cell, given that surface to volume relationship, to generate energy. So how do you do that in going from a small cell to a large cell? You simply can't make the membranes larger. It's insufficient. So to transcend the barrier, the eukaryotic cell needed multiple copies of an internal energy generator like you have in the mitochondria. Because with that folded membrane, you could go from a system like this to having many copies and many more membrane surface, much larger membrane surface area available for oxidative phosphorylation to take place. So this is a difficult, or potentially difficult event, because it required capturing a free-living bacteria and allowing it to replicate, a controlled replication, but allowing it to replicate within the eukaryotic cell. If this hypothesis is correct, then any hypothesis invoking the acquisition of mitochondria by already complex cells would not be supported, because this seems to be an essential step in order to acquire size. As the well-known science writer Nick Lane put it, the underlying principles here are universal. Even aliens need mitochondria. So 3 important points to remind you of. Mitochondria evolved from bacteria. All eukaryotes have mitochondria, and mitochondria are associated with aerobic respiration. And that if we look inside a mitochondria, they have genes. They have 16S ribosomal RNA. It's a different size RNA than we find in the eukaryotes. And that the composition of this RNA is similar to, nearly identical to proteobacteria, as I had mentioned earlier. What about chloroplasts? We're convinced, based on the scientific evidence, that cyanobacteria are the origin for chloroplasts. So chloroplasts evolved from engulfing a cyanobacteria that was again allowed, under controlled conditions, to multiply within the eukaryotic cell. It has its own genome. In fact, if we begin to look at the most important enzyme for photosynthesis, rubisco, part of the proteins associated with rubisco are synthesized by the nuclear genome. In part, are synthesized by the chloroplast genome. And again, if we begin to look at the 16S ribosomal RNA gene in cyanobacteria, it's nearly identical to what we find in plant cells today. So when did all this happen? Well, one way is to take a hypothesis for the tree of life, starting off with Luca. We mentioned that there was a bifurcation of bacteria in one branch, and eukarya and archaea in a second branch. And at this point here, we have the great oxidation event associated with the evolution of cyanobacteria. Someplace along the way, after the great oxidation event, sufficient oxygen became available in the environment that eukaryote captured this protobacteria that we had mentioned before in the Rickettsaceae. Okay? Probably not immediate, because there was a time lag between the great oxidation event and when we suspect that the first eukaryotic organisms evolved. But we have no evidence of that because we have no eukaryotes that don't have mitochondria. At some time later, the eukaryotes captured a cyanobacteria, and that allowed the evolution of plants in this evolutionary tree. So at this point here, we'd have a clicker question. Does the tree here, which is a hypothesis, predict that there was once a eukaryotic cell that did not have mitochondria? And the suggestion here is yes, but the tree doesn't necessarily show that. So Wohl's tree of life predicts ancestors had already diverged before they had mitochondria. The currently available data, however, do not allow us to determine when mitochondria became part of the eukaryotic phyla. We know it had to have happened after the great oxidation event, so after the evolution of cyanobacteria and after the evolution of protobacteria. So we have a sequence, although we might not have the exact years down. Now this is one hypothesis for the tree of life. Another way of expressing that is to arrange the tree of life a little differently. And here you still see the bifurcation with bacteria in one branch and archaea and eukarya in the other branch. But in this expression, we have an inferred time gap when archaea and eukarya had separated, but there might have been a period where there were no mitochondria. As I mentioned earlier, all eukaryotes have mitochondria. We don't know how long this time period was, if it existed at all. So why is a eukaryotic life form useful? One is it allows cells to be larger, a factor of 10 larger. That places a physical constraint on reducing the surface to volume relationship. But it represents an ecological opportunity, one in which natural selection can have a role, because unicellular organisms with these additional membranes have a competitive advantage in terms of energy generation. So a eukaryote that's able to capture and evolve a symbiotic relationship with a mitochondria can generate more energy. Okay? We need this evolutionary solution in order for eukaryotic life and cells to expand in size and importance on our planet.